Okay, so welcome to this next video on the endothelial-derived relaxation factor. Okay, so far what we have seen is that if we give someone an intravenous injection of acetylcholine, what it's going to do is it's going to cause these waves of calcium that are going to propagate through our endothelial cells. Okay, so what we now want to see is what are these waves of calcium going to do? What does a rise in calcium actually trigger within the endothelial cell? Okay, and basically the rise in calcium is going to act through the protein calmodulin. Okay, so let's just have a little bit of a discussion of calmodulin. So the structure of calmodulin is often drawn like this in cartoons. So calmodulin has these two lobes, like so, connected by this linker region between them, which are known as the N lobe and the C lobe of calmodulin. Okay, and each of these two lobes of calmodulin has two calcium binding sites. These are the two calcium binding sites on the N lobe, and these are the two calcium binding sites on the C lobe. Now, these calcium binding sites are what are known as EF hand domains. Okay, so an EF hand domain is basically a polypeptide structure capable of detecting calcium, basically. And I'll show you roughly what its structure looks like. And they are excuse me, they're basically polypeptides. And if this line represents the polypeptide, and they have this loop round, like so. Okay, and this loop is where the calcium is going to bind. So this line represents the string of amino acids joined together, okay? So it's a polypeptide. So if we have amino acid after amino acid after amino acid after amino acid, etc., and you have this chain of amino acids. Now, the amino acids in this loop region here are going to have acidic side chains, basically, so that R groups are going to be acids. And that means that they're capable of donating protons. So the R groups will donate their protons away. And in so doing, they themselves will gain negative charges. So you're going to have a lot of negatively charged amino acid residues in this loop. That is perfect, basically, for coordinating a positively charged cation like calcium. So calcium can therefore come and bind to these EF hand domains like so. Now, EF hand domains rarely occur on their own in proteins. Instead, they often occur in dimers like so. So you have this two EF hand domains linked together by this linker polypeptide region between them. Okay, so EF hand dimers, or an EF dimer is what this will be known as. So these are the structures which you have in the lobes of calmodulin. You have these EF dimers in the lobes of calmodulin. So one EF dimer in this lobe, one EF dimer in that lobe, which means that you have these two calcium binding sites next to each other. Okay, now the structure of calmodulin when it does not have calcium bound to it looks like this. The two lobes are sort of compacted sort of together, there's this sort of curvature here, and this polypeptide that links the two is a linear polypeptide. This structure is known as apocalmodulin. Apocalmodulin. And for short, people often denote this as apo, and then the shorthand for calmodulin is C, then lowercase m, and then capital M. So CAM is the shorthand for calmodulin. Right, so what can happen is when calcium goes up in the cell, calcium ions are going to come and bind to these four calcium binding sites on the apocalmodulin molecule, and that's going to cause a change in conformation of this apocalmodulin molecule. And what it's going to do is it's going to take on a conformation that looks more like this. So the two lobes are going to move apart, like so, so they're going to move further away from one another, and the linker between them is going to also change conformation. It's going to go from being a linear polypeptide to having an alpha helical structure. Okay, right, so this here is an alpha helix, which is a secondary polypeptide structure formed by the backbone of the polypeptide, so the amide link regions. Okay, right, so now what has happened is these calcium binding sites here, one here, and one here, they all now have calcium bound to them. So calcium's here, calcium's here, calcium's here, and calcium's here. Okay, so this is what's known as a calcium calmodulin complex. Okay, and for short, it's often denoted calcium, so Ca2+, and then again you use the shorthand for calmodulin, capital M, sorry. So that's the calcium calmodulin complex. So basically, when calcium goes up in the endothelial cells, you're going to get these calcium calmodulin complexes forming, and those are now going to activate the important enzyme. And finally, after all that sort of preamble, we are now here. We're going to see, finally, the endothelium-derived relaxation factor. And we're going to see, firstly, the enzyme that actually makes it. So, calcium calmodulin complexes are going to come and bind to an enzyme, and in so doing they're going to activate this enzyme, and this basically is going to be the enzyme which produces our endothelium-derived relaxation factor. Okay, and this enzyme, dun dun dun, is NOS, or nitric oxide synthase 3. Now that's the correct name for it. Often it's still referred to as ENOS, which stood for endothelial nitric oxide synthase. Now, that name is not good. The reason is that this NOS3 is found in loads of other places other than the endothelium. So we're trying to phase that name out and replace it with NOS3, but you will still see ENOS. It's a pervasive name among the literature. So, NOS3 then stands for nitric oxide, which is the endothelium-derived relaxation factor, synthase, and then it's the third type of nitric oxide synthase. Right, so, and ENOS just stood for endothelial nitric oxide synthase. So, let's colour this enzyme in because it's so important. Now, this enzyme basically is going to start creating you nitric oxide. So in response to acetylcholine, the endothelial cells are going to activate their ENOS enzymes, or their NOS3 enzymes, and that's going to cause the release of nitric oxide from these endothelial cells. So NOS3/ENOS here. Okay, so basically it's going to start creating nitric oxide, or NO. Now, nitric oxide, the correct name for it is nitric monoxide. So the correct name is nitric monoxide, but everyone just calls it nitric oxide. So this is nitric oxide. Right, so these endothelial cells are going to start producing this nitric oxide, or this nitric monoxide, and what's going to happen is it's going to move from the endothelial cells into the smooth muscle cells which surround the endothelial cells. Let me get my initial picture of the artery back, well, the blood vessel back again. So here are our endothelial cells right at the centre. They're producing this nitric oxide, and what's going to happen is the nitric oxide is going to diffuse through the phospholipid bilayers, because it's a perfectly lipid-soluble molecule. It's going to diffuse through the phospholipid bilayers, and it's going to go out of the endothelial cells, across this basement membrane, and into these smooth muscle cells. Again, it can just cross the smooth muscle cell phospholipid bilayer, and then it's in the cytoplasm of the smooth muscle cell. So our story, therefore, now needs to turn to what it does in the cytoplasm of the smooth muscle cell. Right, okay, so let's turn over and see what it's going to do in the cytoplasm of the smooth muscle cell. Right, so let me just sort my paper out. Right, so let's say we have a smooth muscle cell here, which are these spindle-shaped cells, classically. So here's our smooth muscle cell. Right, so nitric oxide is arriving in this smooth muscle cell. So what does it do? Well, basically, it activates an enzyme known as guanylate cyclase, and specifically, it activates soluble guanylate cyclase. So soluble guanylate cyclase. Guanylate cyclase. So this is guanylate cyclase or guanylyl cyclase, although guanylate cyclase is actually more common here. So this is sort of like contrasts with cyclic AMP signaling pathways, where adenylyl cyclase is probably more pervasive than adenylate cyclase. Here, guanylate cyclase is more pervasive than guanylyl cyclase. So, soluble guanylate cyclase, which we will draw here. So this is an enzyme, so I'll give it its sort of active site there. Okay, and this is often just denoted SGC to stand for soluble guanylate cyclase. Okay, and basically what it's going to do is it's going to make cyclic GMP. So it's going to take in GTP, or guanosine triphosphate, and it's going to convert it into cyclic GMP, or cyclic guanosine monophosphate, and pyrophosphate as well. So it's going to create a PPI, or a pyrophosphate molecule. So let me show you this in a bit more detail with a cartoon below. So, the structure of guanosine triphosphate then. You have, I'll draw it here. You have a ribose sugar here, so here's your ribose sugar. Then you have this guanine organic base, which I'll just denote as a box because it would be far too much effort to actually draw the structure of that. Then you have the 5th carbon coming off here, and then off this you have 3 phosphate groups like so. So that is guanosine triphosphate, or GTP. So let me colour in the different bits of it. So these are the phosphate groups in pink. So you have 3 phosphate groups, hence the triphosphate. And here is the ribose here, which I should highlight that 5th carbon up there as well because that's part of the ribose. And finally, the organic base is in pink. So the organic base, guanine, and the ribose sugar, together, those are known as guanosine. So guanine is the organic base, but guanosine is the organic base bonded to ribose, basically. It's what's known as a nucleoside, which just means a ribose sugar bound to an organic base. Okay, then you've stuck on 3 phosphate groups, hence its name, guanosine triphosphate. Now basically what soluble guanylate cyclase is going to do is it's going to convert this into cyclic guanosine monophosphate. So let me show this. So again, the guanosine remains the same. So here's the guanosine, the ribose sugar with the organic base guanine here. So let's highlight the different bits in. So here's the guanine organic base again in purple here. Okay, and then in blue, the ribose sugar is here. Right, so what changes is you cleave 2 of these phosphate groups off to make a pyrophosphate molecule over here. So here's pyrophosphate, which is just 2 phosphate groups linked together. Okay, and then finally, the final phosphate, now it gets linked to this 3rd carbon of the ribose sugar down here. Okay, so that there is why it's called cyclic guanosine monophosphate. It's guanosine with a single phosphate, but it's not just a single phosphate sticking up here. It's formed this cycle, basically, because it's linked to the 3rd carbon. So this is cyclic guanosine monophosphate, or cyclic GMP for short. Right, and it's basically exactly the same as cyclic AMP, except that it's got the guanine organic base here. So this is cyclic GMP. And this here is the pyrophosphate, PPI. Okay, right, so that's the reaction that this soluble guanylate cyclase is catalyzing. Okay, so what does cyclic GMP do? Well, basically, it activates an enzyme just like cyclic AMP activates an enzyme. And it basically activates the enzyme cyclic GMP dependent protein kinase. Or it's got another name, protein kinase G, which I prefer to cyclic GMP dependent protein kinase. Okay, so this is a kinase enzyme, so I should draw it with a little active site there. Okay, so protein kinase G, this enzyme is here. This is protein kinase G. Right. And its other name, which you will see occasionally in the literature, and even Wikipedia uses this other name, cyclic GMP-dependent protein kinase. Okay, so cyclic GMP-dependent protein kinase. So both of those names, they're referring to the same thing, basically. This enzyme, which is a serine threonine kinase, which is going to be activated by cyclic GMP. Okay, so far in this story then, we have had nitric oxide released by the endothelial cells, and that, by the way, is the endothelial-derived relaxation factor. This is what EDRF is, endothelial-derived relaxation factor. It is nitric oxide, and so far what we've seen is it's going to activate this enzyme, protein kinase G, in the smooth muscle cell. In the next video, we'll see how protein kinase G is going to cause relaxation of the smooth muscle cell.